Swayam Prabha. Digital India. Educated India.
Good day everyone, welcome to the course on next generation sequencing technology, data analysis and applications. In the last few classes we have discussed several mapping algorithms, and in the last class we looked at the BWT based mapping, right, and we have seen that this is the method that is probably the ideal method for us because it is fast, it does not take too much memory, and it can handle all sorts of reads, right. It can handle reads with mismatches, and it can also map reads to multiple positions or mapping to repeat sequences. So, what we are going to do today is we are going to use a tool, right, and look at a tool that utilizes this BWT, and we will do this hands on, right. We will do this read mapping in this class and the subsequent class. Okay, so let us start with that. So, the tool that we will discuss is called Bowtie2 tool, right. So, this will be the concept that we will be talking about, right. So, we will talk about Bowtie2 tool for read mapping. Okay. So, how does this work, right. So, we have talked about this, and just to remind you, right. So, we have seen how we generate BWT through string rotations and lexicographical sorting. We have talked about FM index in the last class, right. It consists of BWT and additional data structures, and what we have seen is with some examples is that read mapping is fast, scalable, and it does not require too much memory, right. And if you want to store the BWT, you can also store this without too much space, and it also, this mapping works very well for reads with mismatches, and if you are mapping reads to repeat sequences. Okay, so, there are several tools that are available for this that utilize this BWT for mapping, right. So, you have Bowtie or Bowtie version 2, which is the Bowtie2. You have BWA and its variants called BWMM or BWASA. So, the tool that we will be discussing is Bowtie, its version 2, right, and this was published in 2009. This is the paper where they actually describe this method, right. So, what are the features of this tool Bowtie2, right. So, we will describe this and look at the tool little bit, and then we will go and install this, right, set this up, and we will do the mapping ourselves, right. So, what are the features of this tool? So, it utilizes this FM index LF mapping with backtracking. So, which means it will allow you to map reads with mismatches, right. And it also uses double indexing, right, to prevent excessive backtracking. So, it is called forward index and reverse index, right. So, you will see these index files when you actually go and do the hands on. And there are of course checkpoints for storing ranks in the BWT, right. So, we have discussed this in the last class. So, if you want to store all the ranks, that will take too much space in the memory, right, or if you want to store them in the hard drive, that will take too much storage, right. So, you need to optimize this, and you can decide how often you store these ranks, and so this method also utilizes these checkpoints. So, if you compare performance of Bowtie2, right, you can see this, so published by the authors themselves, right, against two tools, one is SOAP and another is MAC, right. So, these are again we have discussed the other methods. So, SOAP uses the hash table based methods, and MAC utilizes something like creating index from the reads and then searching through the reference, right. So, what you will see here, right, on the left you have the names of the tools, right. So, here in the first set there is a comparison between Bowtie2 version 2 versus the SOAP, right, and the platform is server, and you can see the CPU time or wall clock time, right. So, the time taken to do a certain number of mappings, right, and you can see Bowtie2 is significantly faster, right, much faster here, right. So, you can see it took about 15 minutes, right, to map those reads, whereas SOAP actually took 91 hours. Okay. So, you can see the difference, right, and you can also see this column here, reads mapped per hour in millions, right. So, Bowtie2 actually could map 33.8 million reads per hour, whereas SOAP actually mapped only 0.1 million reads per hour, right. So, there is a substantial difference in the speed. Also, you can see the virtual memory, the memory footprint in megabytes, right. So, how much memory it is taking. Bowtie2 took about 1.2 GB, right. So, it's 1.15 MB, right. So, about sorry 1150 MB. So, it's about 1.2 GB, whereas for SOAP it's much higher, right. So, it's around 14 GB, right. So, that's a significant difference, right. That means you can run Bowtie2 in your desktop, whereas you cannot run the other program in your desktop computer, right. So, you can see the speed up, right. So, Bowtie2 was much faster, and also there was no difference in the percentage of reads that were aligned, right. So, that means the performance is not the output, the results, there is no difference almost, but this method Bowtie2 is much faster, and it also takes much less memory, right. So, similarly there are other comparisons in Bowtie Mac, right. So, you can see in PC, in server versions, and in all these comparisons you can see these differences, right. So, there is substantial difference in speed between Bowtie and Mac, right, and you can also see the memory usage, right. So, memory usage in Mac is slightly lower, but where that is there is a trade off, right. So, you actually utilize more time, you need more time, right. So, both of these are manageable, right. So, 800 MB and 1.4 GB, that is not too much of a difference, and you can run them in the desktop computer, whereas Bowtie excels is like it is actually much faster, utilizing slightly more RAM. Okay. So, again the performance was not all that different, right. So, Bowtie could map about 72% reads, whereas Mac actually could map about 75% reads. So, all right. So, what we are going to do now is we are going to look at Bowtie2, right. We will set this up, right, and we will start look at the functions, look at the options, and we will also run them to actually generate our own mapping. Okay. So, let's go ahead with that, right. So, here is the link from where you can download the Bowtie2 tool. It is a free tool that you can download and install, and here is a detailed manual, right, giving all the instructions that are required for running this tool. Okay. So, what we are going to do is now we will go to this link and we will download and set this up for our system, and then we will run Bowtie2 to map our reads. Okay. So, let's go there, right, and we will talk about all the options that are there. Okay. So, if you go there, right. So, if you go to this link, right, here is Bowtie2, you can see, right, and it gives you some details, right, some descriptions about Bowtie2, right, and what you will find here, if you come down here, right, it is Bowtie2 version 2.5.1, right, and if you go to this link, right, you can download the latest version, which is a zip version, or you can also look for your specific system specific version, right. For example, if you are running Mac, you will probably want to install this one, right. So, you can download the version appropriate for your system, right, and then you can work with that system, right. So, what I have done, I have downloaded the latest version is this one, right, and I have stored this, right, otherwise it will take a bit of time. Okay. So, I will now go into that folder, right, where I have actually stored this downloaded version, right. So, here you see this here, right. So, this is the Bowtie2 latest version, and this is the zip folder, right. So, what we will do now, we want to unzip, right. So, hopefully you remember these commands, right, that we have discussed, and we are in that folder where we are doing all these operations. Okay. So, first let us look at this. So, here it is. So, if with ls we see this Bowtie program in the zip format, and to unzip this we simply write unzip Bowtie .zip, right, the name of the file. So, this will create this unzipped version. So, you can simply clear the screen and then again look at this, and you will see that there is a folder now with this Bowtie2, right. So, this Bowtie2 folder contains the programs that we are going to use. Okay. So, we can go in there and see the programs, right, and here are the programs that we can use for our mapping. Okay. All right. So, the setting up, as you have seen, is very easy, right. There is no complications. You just simply download the zip file, you unzip, and in your system you might have to change permission, but here I think the permission is there. So, we can also check the permission, right, by saying ls minus -l, right. So, for these programs, especially this Bowtie2 L, right, Bowtie align L, Bowtie2 align S, etc. These are the executables, right, that will run when you want to map reads. So, you want to make sure that these files, you have this execution permission, right. So, here in this system, right, we have this execution permission X for all of them. In your system it might not be there. So, you want to make sure that you have this execution permission. Okay. If you have that, and that is it, right, we are actually ready to go. Okay. We can utilize this and run the mapping. Okay. All right. So, we will come out, right, and we will be in the test 1, right. So, now one of the things we will discuss is how we actually run these codes, right. So, there are different ways to do this, but I will go back and I will show you some of these steps. Okay. Okay. So, how do you actually run this? So, we will also look at the manual, right, once we actually start running these commands. Okay. So, it appears that so this is a two-step process, right. So, you would have to first build something called a genome index, right. What happens in genome index is actually this Barrow-Siller transform, this FM index creation, double indexing, and checkpoint creation, right. So, all these things are happening when you run this genome indexing part. Okay. So, that is the first step. Okay. You would have to do this, and once you have done this for a genome, right, you can use that for all sorts of mapping, right. You do not have to create that index again and again. Okay. So, once you have created that index, you can simply store it in your hard drive, and you can use this whenever you are mapping reads from that organism. Okay. So, this is the first step. Okay. And there is a different command to do this, and in the next step, once you have built this index, you do the read mapping. Okay. So, there are two different commands which we will see in a moment. We will also use those commands for our purpose. So, the first one is the first step, right, is the building genome index using Bowtie2. The command is Bowtie2 build. Okay. So, this is a specific command to build these genome indexes. Okay. So, I am showing you the how you should write this command in the terminal, or how you should run this command in the terminal, right. So, this is Bowtie2 build followed by certain options which might be useful in certain cases, followed by this reference IN. This is the list of reference genome sequences, right. It could be a single reference genome, or it could be multiple reference genomes, right. So, maybe you have sequenced one reference genome along with certain other fragments, then you want to use this multiple reference genome to build the index. Okay. And then you have BT2 base, means you want to give the base file name for the index file. Okay. So, what it will do, right, when you run this Bowtie2 build, we will see is that it will create these index files, right. So, containing this all this information, FM index, etc. right. So, this BT2 base file will then be useful for the mapping process, right. So, you want to specify the name that we will use again for the mapping process. Okay. So, what are the options? So, what are the explanations? So, as I mentioned, reference in is it's a comma separated list of files containing reference sequences. So, it could be just one single file, or it could be multiple files, okay, which should be comma separated, right. So, there should be a comma between these files. Okay. And then you have this BT2 base, which is the base name of the index file, which will be written in this .bt2 format, right. So, you will see when we actually run this, we will find this out. We'll see these files created after we run this command. Okay. Now, the question is, how do you find the reference sequences, right? So, you might be wondering, right, okay, great, reference sequence, how do you find this reference sequence? Okay. So, there are places where you can find reference genome data, right, and that's stored in this NCBI link. It's a good resource for finding this reference genome data, right. So, we'll have a look, right, where you can actually find this reference genome data very quickly, right. So, if you go to this link here in the RefSec, it's called the RefSec, right. So, this is the reference sequences, and you can simply go to RefSec FTP or RefSec genomes FTP, right. So, maybe I zoom in bit, right. So, if you go back, right, here is the link, right, RefSec access. Here you have RefSec genomes FTP, where you will find these reference genome sequences, and they are organized by different classes, right, archaea, bacteria, fungi, invertebrate, etc., or plants. So, you can go and click any of these links, right, and you can find list of species, right, for which you will have the reference genome data. Now, for reference genome, right, so when you click on these links, you go inside, what you will get is something like latest assembly versions or representative, right. So, representative ones will be giving you the reference genome, and what you need is the FASTA format file. Okay. Remember, for running this Bowtie2 indexing, it is preferable to have the FASTA file, right, and you have to search for the genomic FASTA. Okay. So, here you will find some of these, right. So, for example, this .fna, this is the FASTA file, or .fa, that is the FASTA file, right. So, here this is the FASTA file, right. So, this fna.gz, right, you can see this, right. So, this fna file, this is the FASTA file, and it contains the genomic sequence, and it is just compressed, right, with gzip compression. We have learned about this gzip compression, right. So, in our case, the data that we have worked with so far, it comes from yeast species, right. So, let us go and find the yeast genome, right. So, here we will go inside, right, and we will find this, right. Let us see. Okay. So, here it is. So, the yeast species, right. So, here is the fungi, right, and we have Saccharomyces cerevisiae, right. This is the genome that we need, reference sequence, and you can see reference here, right, representative or reference, and reference sequence we have here, right. We just click on this, and you have lot of files, but what we need is this genomic.fna. Okay. So, you can simply click on this and you can download, right, and you can then utilize this file for building the index. Okay. You would have to z unzip, right. So, because this is gz compressed. All right. So, what I have done, I have actually downloaded this file already, and I have this in my system. Okay. So, let us now look into let's now go back into this slides, right, and look into the options, right. So, we have learned about this Bowtie2 build, we have understood the reference file, where this reference file will come from. We have talked about BT2 base, this is something we have to specify, where the index file will be created, right, the name of the index file, and then you have something called options, right. So, these options could be useful in some cases, so that it is good to know what these options are. Okay. So, for example, the options that you can give is this -f means the reference input file, these are in FASTA format. Okay. So, this is the default option, but you can also specify, okay, we have FASTA files. So, there is another option which is -c, which actually you will specify when you are giving the reference sequences in the command line itself instead of the file names. Okay. So, if you are writing this command Bowtie2 build, right, instead of reference in file names, right, instead of file names in reference in here, right, what you do is, instead of reference in files here, you put sequences here. Okay. But this is quite rare, right, because these sequences are so huge, we would not put them in the command line, right. So, we will usually put them in files and then direct to the files from this command line. Okay. There are other options. So, for example, you can use this minus threads with a number followed by a number. So, this int means there should be an integer, right. So, if you will say for example, minus threads 4, right. So, it is kind of parallelize the process. So, it will actually speed up the index building process, right. So, this is something to do with the computer system. Okay. Right. So, once you have built the indexes, right. So, then you can go ahead and map breeds with Bowtie2. Okay. So, in this class, in the remaining part of this class, what we will do is we will actually start looking at this index building process, right. We have downloaded the reference genome. So, what we are going to do now is we will look at the commands options and we will build the index, right. So, let us do that. Let us go to the command line, right, and we will actually see this process in action. Okay. So, one of the things you will probably find out, so one of the things you will probably find out here, right. So, we have the reference genome in this folder. So, here is I have stored the East reference genome, you can see, right, and inside that this is where the file is. Okay. So, I have a lot of these files, but the one that we need is this one .fs. Okay. S28HC reference sequence R642 1.fs. This is the reference file, right, again in FASTA format. So, FASTA format file, the extensions could be different, right. You can have .fa, .fsa, .fna, right. You can open up those files and you can simply check, right, whether these are in FASTA format, right, or you can simply write head, right, and you can see whether the files are in FASTA format. Here you can see this is the header line starting with this greater than sign, right, and you have the organism name, Saccharomyces cerevisiae, and then from the next line you have the sequences. Okay. So, this is the FASTA file that we need for building the index. Okay. So, we have the reference file. Now, we want to see how we will run this Bowtie2 command, okay, Bowtie2 build command. Okay. So, we have installed this Bowtie2 in this folder, we have extracted, and inside this folder we have all these commands. Okay. And there is this command Bowtie2 build. Okay. So, if you search simply inside this, right, you will see this command here. You have Bowtie2, only Bowtie2, you have Bowtie2 align, and then you have Bowtie2 build, you have other options Bowtie2 build L, etc. Right. So, this Bowtie2 build command is what we require. Okay. So, what you can do, right, you can call this command by typing the path, right. So, this is the folder in which this command this function resides, right. So, you have to specify the path, and we are using this dot beforehand because just to denote that this is an executable. Okay. So, dot slash Bowtie2 build, and you can simply run this by saying minus help. Okay. So, what will happen if you run this with minus help? It will actually show you all the options. Okay. It is taking a bit of time, I do not know why. Okay. So, it is here and it tells you this Bowtie2 version, the author name, etc. Okay. And the next line is very important. Okay. So, have a look carefully. Okay. So, please check this line because this gives you the usage information. Okay. So, what it means, how you should use this command. Okay. So, usage is Bowtie2 build, right, then the options, and then the reference sequence, then the BT2 index base, right. So, the name of this index file that you have to specify, right, and we have discussed this reference in BT2 index base, and then the options are given here, specified here. Okay. You can see these multiple options. We have discussed only some of these options, right. For example, we have discussed -f, we have discussed -c, but we have not discussed the other options. So, these are not really very important, but you can of course go through, and if you see some of these would be useful in your case, you can use them, but what I can assure that these are probably not very much required for daily use. Okay. So, we can just use this minus A for minus C, right, or minus threads, right. This is the most important one, because we can speed up the process if you have a fast computer where you can run multiple threads, or in servers, for example, we can use these multiple threads. Okay, so this is how this whole process will work, right. So what we can do now is we can follow this, right, and we can run these commands. Okay, so remember now, right, sorry, this one here. Okay, so this is the command Bowtie2 build, right. We are calling this. We can give options, but let us skip options. We do not really need that. What we need, so we have given space, then what we need is the path to the reference file. Okay, so where is the reference file? Let us give that path. So it is inside this folder, right. In your system, it will be the path where you have stored this downloaded file. Okay, so that's where you should point. Okay, and this is the .fsa, right. This is the reference sequence .fsa in FASTA format, and the BT2 index name, right. So that base name we need, and I will simply write this as yeast whole genome. Okay, so we are creating this index for whole genome. So why I am saying this whole genome, because sometimes you will see for some organisms you can also find reference transcriptome, which is different from the reference genome, right. So you can probably understand what is reference transcriptome. Is the list of, or the sequence of all mRNA, right, that are expressed. Okay, and that will be different from the whole genome reference sequence, right, because the mRNA expressed, that will be just part of the genome, right. There won't be any intron, right, the non coding regions will not be there, right. So sometimes you will find that for some organisms you have reference transcriptome, and you want to use that reference transcriptome in, if you are mapping, for example, RNA sequencing data, right. So in case of transcriptome analysis, you will generate RNA sequencing data, and in that case you will want to build a index for reference transcriptome, right, and then you will map against that reference transcriptome using those indexes, right. So why do you want to build these two separate indexes, right, for genome and for transcriptome if available? Is that because the search against transcriptome will be much faster, right. The transcriptome contains a much smaller fraction of the genome, right. So the index that will create will be much smaller in size, and the search process, the mapping process will be much faster. Anyway, back to this. So I am naming this as yeast whole genome, right, because this is the genomic reference sequence, and we will simply press enter and it will run. Okay, so it might take a few seconds, right, or a few minutes to run, and it is working, right. So you see, like it will give you a lot of this process, of course not so important really. It's kind of creating these indexes in stepwise manner, right, and finally it will actually generate those indexes, right. So okay, so you can see, right, this is going on, this Barrow-Wheeler transform, the FM index calculation, the double indexing, the checkpoints, right, all those things were created here. Okay, so what you have now, you have these last commands, you can see, right, you can see this, right, you have these .bt2 files. Okay, we can see these bt2 files also, right, here. We can simply say ls, right, and we will see multiple bt2 files that have been created here. So you have yeast wg.1, wg.2, wg.3, etc., and you will also see some of the reverse indexes, right. So you have this wg reverse.1, right, reverse.2, and this is the double indexing that we have talked about, right. So by the way, just if you want to just see these bt2 files, right, in your system, rather than checking all the files, you can simply say ls star dot bt2. So what it means is that star means wildcard, right. So it will match anything that it finds. So it will just show all the files that end with this .bt2, okay, all files in bt2 format, okay, and simply you will find these 6 files created in your system. Okay, So, once you have created these files, right. So, this is the first step that is now complete. Okay. We have built this index, and you can keep these BT2 files for as long as you want, as long as you are working with this genome. You do not have to recreate these BT2 files unless the reference genome sequence is updated by a newer version or so. You do not need to work or change these reference indexes anymore. Okay. So, once you have created them, you can just keep them forever, and whenever you want to map reads against this Saccharomyces cerevisiae genome, you can utilize these indexes, right. So, it's a good idea, right. Once you create, you can keep this, and they do not take too much space, right. So, you can also look at their space usage, right, and you can see they are not taking a huge amount of space here in the system. You can compare this, right. So, if you can compare this to the read sequence data, right, and they are actually quite okay, right. All right. So, going back to the presentation, right. So, we have now created the indexes, right, out of this reference sequence using this Bowtie2 build command. The next step is actually the mapping process, the actual read mapping process, and we will discuss that in the next class, because this is a very elaborate step, right. We have lot of options and some of these options are very useful for mapping. All right. So, here are the references for this class, and to summarize what we have seen is that Bowtie2 is a very fast read mapping tool. So, this is something we have discussed, and it requires less than 1.5 GB memory for working with human reference genome, right. And Bowtie2 is run in 2 steps, right. So, we have the first genome index building step, and then you have the actual mapping part. So, we have in this class we have actually done the hands on where we have actually looked at the genome index building part, right. We have not looked into the mapping part, which we will be doing in the next class. And what I have shown is that Bowtie2 build this the function that actually builds these index files, and you can store these index files once you have created them, and you can utilize them for mapping reads of that organism in future. That is it. Thank you.